So thank you very much for this honor to be here presenting on the updates on some of the new developments that have occurred since the last Sangal meeting a couple of years ago. I'm going to focus on one of the targets that we feel is very exciting, and that's the PI3 kinase itself, and also the PI3 kinase pathway. In the next 20 minutes or so, I'm going to give an overview, and it's listed here. I'd like to review very briefly the pathway so that we are familiar with it. Some data that supports that the pathway, the PI3 kinase itself, may mediate lack of response to upstream HER2 therapy. Some proof of concept that targeting this pathway is a useful approach, and we're going to focus very briefly on the mTOR data. I realize that the talk by Dr. Ken Osborne is going to be also dwelling on this topic as well. Then some clinical trial results looking at pan-PI3 kinase and the isotype-specific alpha inhibitors. And then finally, some thoughts on some future directions that hopefully, and if invited, we can discuss in the next Gallen meeting when we will have more data. The PI3 kinase mTOR AKT pathway is a critical pathway in breast cancer. It's in the middle of the signaling process of receptor tyrosine kinases, and that includes HER2, HER1, HER3, FGF receptor, IGF-1 receptor, PDGF receptor, etc. And downstream components of the pathway, such as mTOR, together with the signaling of nutrients and oxygen and other key components in the cellular component, they determine the fate of the cell. It determines whether the cell will proliferate or not. It determines whether the cell will grow. And also, it controls angiogenesis and apoptosis. So in all, very, very critical. Now, there are several strong evidences that this pathway is important in breast cancer. To begin with, is very frequently mutated. So PI3 kinase itself is mutant in about 40% of breast tumors, depending on how deep do you look, and is also a mutant in about 25% of HER2-positive tumors, less so in the triple-negative, about 8%. So that's first proof of evidence. Second proof is that PI3 kinase mutations itself are transforming in experimental models. So if we put all this together, if we put together the fact that it is transforming and the fact that it is very frequently mutant, I think it makes a very strong rationale to try to target it. In addition, there is proof that this pathway that is downstream from HER2, when mutant, results in a poor prognosis in patients with HER2-positive disease when treated with HER2 antibodies. You may be familiar with the Cleopatra study. That's the phase 3 study in the first-line setting that led to the approval of pertuzumab in patients with HER2-positive advanced disease. And you have the design here. What we did here, I'm not going to talk about Cleopatra. What we did here is that we put together all the patients in this study. They all received some form of HER2 therapy, either trastuzumab or the combination of trastuzumab and pertuzumab. And we asked the question, are there any prognostic indicators of lack of response? And there was a very comprehensive analysis. We looked at many parameters. PI3 kinase mutations conferred resistance to the HER2 therapy. And here what you see is that the wild-type tumors do better, which is another way of basically mentioning the same data. And if we look at the Kaplan-Meier, then it becomes even more apparent. This is the control arm. These are the patients that were treated only with trastuzumab. And what you see is that those that have the mutation, that's the lower line, have a poor outcome as compared to those that have wild type. And the same applies to the tumors that to the patients that were treated with pertuzumab and trastuzumab. Again, the PI3 kinase wild type do better than those with the PI3 kinase mutation. So I think that sets the stage clearly to try to combine anti-HER2 therapies with anti-PI3 kinase therapies if we want to improve these curves. And that's something that is an area of clinical research. Now, the proof of concept was established this year by the data that was obtained with mTOR inhibitors that, as you have seen, mTOR is downstream from PI3 kinase. And there's a lot of evidence that there is crosstalk between mTOR and the estrogen receptor pathway. So what happens is that mTOR has the capacity to activate estrogen receptor transcription in a ligand-independent fashion. And even if we block an ER-positive breast cancer with an AI inhibitor, what we are doing is that we are preventing ligand-dependent ER transcription, but we are not preventing ligand-independent ER transcription. And the converse is also true. If we just block with the mTOR, we have a hyperactivation of ligand dependence. So I think there is a lot of evidence that supports that we need to combine these two therapies in ER-positive breast cancer. And this is exactly what we did. This is the Bolero 2 study design, large study that was a randomized registration study, 2-to-1 randomization in patients that had received already one line of an AI with letrozole or anastrozole. And the study, as you are familiar, was quite positive with a hazard ratio of 0.36. This is probably the most positive trial in metastatic setting in patients with ER-positive disease, or for that matter, with breast cancer that showed this improvement, and also other parameters that we looked at, such as bone disease, etc., that resulted in a better outcome. So this is exciting, and that's the first proof of concept. Now, I think we can do better, and we can do better with the new class of agents which are targeting the PI3 kinase gene itself. So this is a list. This is a partial list of the inhibitors that are in clinical trials. I say this is partial because these are only the compounds that are being studied at our institution. So there are many others. The compounds are divided by the subtype of the PI3 kinase gene that they being targeted. So as you know, there are 4 isotypes: alpha, beta, delta, and gamma. Delta and gamma are extremely important in lymphoid malignancies, and you may be familiar with some really amazing data of delta inhibitors in patients with lymphoma with major responses. And alpha, the alpha subtype, is the unit that is mutant in breast cancer. So we have compounds that just hit the alpha subtype. These are the ones in the top. I'll talk to you about this in a minute. And then we have compounds that hit the whole 4 subtypes. These are what we call the pan-PI3 kinase inhibitors. And then down the list, we have dual PI3 kinase mTOR inhibitors, catalytic mTOR inhibitors, as opposed to the rapalogs that just inhibit mTOR1. And then finally, and I will not touch this today, we have the AKT inhibitors that probably have a different indication. I think the AKT inhibitors are probably going to be better in tumors that are triple negative, because in preclinical models, they work far better in tumors that have PTEN loss as opposed to PI3 kinase mutation. But let's focus for a second on the pan-PI3 kinase inhibitors. This is data from Dr. Meyer at Vanderbilt University. You may be familiar that there is a stand-up to cancer group that is addressing the issue of PI3 kinase targeting in breast cancer. And as part of this initiative, a number of clinical trials are evolving. And one of them is the combination of this compound, VKM 120, with letrozole in ER-positive metastatic breast cancer. And this is a busy slide, and I would not like to go into much detail. But if you can just look on the here on the table on the left, you will see that we saw clear-cut responses in this combination. So it is feasible, it is well tolerated, and based on these data and the strong preclinical data also supporting this approach, we have launched a major study. That's the BEL-2 study. That's a registration study with a combination of fulvestrant plus VKM versus fulvestrant and placebo in patients with advanced disease. You will notice some interesting aspects in this study design. For example, we felt that it was important to be able to stratify patients based on the PI3 kinase mutational status. We need to know whether these PI3 kinase inhibitors are more efficacious in tumors that harbor PI3 kinase mutations. Yet, if we do a study like this, that has more than 300 sites. I think that many of the sites don't have ready yet the routine analysis of PI3 kinase mutation as part of their clinical care. So I think a smart study design that has been also done, is being done also in other studies, is that patients enter into the study. We have a run-in period in which they receive fulvestrant, and in the meantime, we have the tumor in our lab, not in our lab, in the central lab of the study, and they are looking for PI3 kinase mutational status, and by day 15, when we know the mutational status, then patients are randomized to receive the fulvestrant plus BKM plus the fulvestrant and placebo. So this is ongoing. As of last week, we had 150 patients in the study. It's enrolling very well. We don't have any safety signal. And I am hopeful also that the US sites are beginning to catch up. So we should enroll this trial even way ahead of schedule. Very exciting. Now, if we focus on the alpha inhibitors, these compounds came earlier, so development is less advanced. But I'd like to point your attention that this could be perhaps a better approach, and we don't know, but I think it could be better in patients that harbor tumors with alpha mutations. Why is that? Well, look at the selectivity. In this graph, you have the selectivity displayed by alpha, wild type, and then the alpha mutations, very, very low nanomolar concentration to inhibit an IC50 as opposed to the other subunits and as opposed to mTOR. So that's one thing. The second thing that to me is even more important is that Novartis and also a collaboration between the Sanger and MGH, we have a robotic platform in which we check hundreds of cell lines, close to 1,000. This was published in Nature in back-to-back papers this year, over close to 100 cell lines that through a robotic screen, they are checked for hundreds of compounds on a continuous basis. And in a totally unbiased fashion, the computer analysis makes correlations between outcome and a given mutational profile. Well, if we do this with this compound, here you have on your left the sensitivity on the cell lines that are PI3 kinase mutant. And you will see that 74% of these cell lines, which is a lot, are sensitive to this inhibitor, as opposed to only 30% of the cell lines that are wild type. So based on this data, we embarked on a first-in-human phase 1 study in which every single patient that came in from first day had an alpha mutation in their tumors. And what we observed, and that, again, that's a phase 1 dose finding. We have patients here that received different doses, but what we found is a very nice response, as you can see by this waterfall plot. And in the case of breast cancer, we saw solid responses that are displayed on this slide. This is vastly different from what we have seen with the pan-PI3 kinase inhibitors, and vastly different from what we observed with our mTOR inhibitors. So we are very hopeful that this will be more active in this patient population. And I am talking about solid responses. This is a patient of ours that we treated at MGH and shows a beautiful response. So we're not talking about small responses here. And we have another compound that is, this one is a beta sparing, but is mostly an alpha inhibitor that has the same profile. This is showing a panel of cell lines, and what you have in the bottom in the red squares is the mutational status of the cell lines. So those that have the red box, they harbor a PI3 kinase mutation, and you can see clearly that these cell lines are those that are sensitive to this compound. And not only that, but in the mutant cell lines, what we see is a profound induction of apoptosis upon therapy with this compound. We will present the clinical data at AACR. I cannot disclose you the findings, but suffice it to say that it's going to be a major plenary presentation, and it's for you to guess whether the study shows activity or not. So what do we know today? I think what we know is that we have seen clearly high activity in these mutant breast cancer cells, and that we need to study clearly additional determinants of resistance and sensitivity in these patients. And then what's very exciting is the interaction with the estrogen receptor that we are observing. So then moving to the next clinical leads, and that's the ER story. We have done multiple combination studies with anti-estrogens, and we see every single time a tremendous synergy when we combine anti-estrogens with PI3 kinase alpha inhibitors. And this is a busy animal experiment. The point that I'd like to show is that the bottom line that you see here, what it has. Oh, this is working. Okay, what you see in this line, is this working? Or, well, I don't know what I'm doing. In the bottom line, you see the combination arm, measure anti-tumor activity. But then we have two arms in which we begin with one therapy and then we add the other, and we also see a tremendous activity. So the combination is very exciting. Doing gene expression analysis, what we see, and that's something that is observed around here, in this part here, what we see is a profound induction of ER gene expression, and we confirm this with Western blot. So that's exactly what we suspected. You block PI3 kinase and you increase ER signaling, and that's the need to block the two together. And that's what we're going to be doing a number of clinical trials with this. I'll just go over. There's a trial that we're doing a memorial addressing this combination. And then, if I may, in the last 3 minutes of my talk, one last story that is very immature, but that is quite interesting. In the old literature, sitting in obscure papers, there was data that suggested that PI3 kinase was regulating DNA damage in triple-negative breast cancer. It was there. It was not looked in detail, but it was present. So we were attracted by this as part of the Stand Up to Cancer group, and we showed that if we interfere with PI3 kinase function, either by short hairpin interference, as shown on the left, or by, as shown on the right, by BKM120, we have a measured induction of DNA damage as measured by gamma H2AX assay, as you can see here. And what we have found is that PI3 kinase inhibition results in a profound decrease in the levels of BRCA1 and BRCA2. So in a way, PI3 kinase inhibition renders the cells functionally BRCA null, deficient, if you wish. And that basically mimics the same situation as the BRCA deficient tumors that are sensitive to PARP inhibitors. So based on this, we conducted a lot of preclinical studies combining PARP inhibitors with pan-PI3 kinase inhibitors. We see very strong synergy, and the Stand Up to Cancer group, based on this data, is doing a clinical trial. At the same time, Luke Canley, in a different model, in a model that is BRCA mutant, he also showed the same effects. So it's all very confirmatory, and I think that supports clearly to moving forward with this approach. So in summary, pan-PI3 kinase inhibitors have shown clinical activity and are under late clinical development in combination with anti-estrogens. The alpha-specific inhibitors have shown remarkable clinical activity in PI3 kinase alpha mutant breast cancer. PI3 kinase inhibition results in the compensatory activation of ER levels and transcription, and that sets the rationale to combining PI3 kinase inhibitors and hormonal therapy. And finally, in triple-negative breast cancer, PI3 kinase inhibitors then regulate BRCA expression and are synergistic with PARP inhibitors, and clinical trials are ongoing. I'd like to finalize by thanking our group and our collaborators. And this is a picture of our new building where we have our new research laboratories. Thank you very much.